And I'm going to introduce Dr. Mike Wimberly is a professor in the Department of Geology and Environmental Sustainability at the University of Oklahoma. He's the head of the Ecograph Research Group. His research combines ecological models with Earth observation data to address scientific questions and to create practical applications in the fields of public health and natural resource management. He holds a BA from the University of Virginia, an MS from the University of Washington, and a PhD from Oregon State University. So without further ado, it is my great pleasure to present Dr. Mike Wimberly.
All right, so without further ado, so what I'm going to talk about is, I'm going to start by giving kind of a quick introduction, talking a little bit about land cover and its relationship with mosquitoes. And then I'm not going to go too deeply into the technical details on anything, but I want to give you some vignettes on some research that I've done in a variety of different places on some studies of mosquitoes in Sioux Falls, South Dakota, land cover and malaria in Ethiopia, potential dengue transmission in the US in relationship to microclimate. And then, for some of you who may be engaged in this type of research or interested in undertaking it, I have just a few general thoughts on analyzing mosquitoes and land cover data to close with. So let's start and maybe think about land cover a little bit. And like a lot of geographic phenomena, one of the best ways to start thinking about and understanding land cover is to just look at it. So these are some data from Northwest Atlanta, Georgia in the US from the National Land Cover Database. And you can see a variety of patterns here. The reds are developed areas, cities, buildings, roads. Green are forests. Yellow is pasture hay. Brown is cultivated crops. So we can see, we're kind of on the edge of a big metropolitan area. We have kind of an outlying city where you see that black box. We go from urban with pockets of islands of forest in the southeast to a more rural landscape in the northeast. So, these are when we think about mosquitoes and land cover, we're asking questions about, okay, how does this variability in land cover potentially influence the distribution of mosquito species and the probability of disease transmission? Let's zoom in for a closer look. Let's take a look at that box. So here I have, again, on the right, this National Land Cover data set. These are, again, classes from a satellite called Landsat. So this takes basically 30-meter pixels, boxes that are 30 meters on a side, and puts them into one of these categories that I showed you here. And then if you look on the left, this is, it's a very high resolution satellite. So we can actually see in true color more of the details of what's on the landscape. So, if you kind of look from left to right, it kind of makes sense. We see a lot of bright buildings and pavement, and that's where the red is. Trees are where the green is. But, we see some other things too, so for example, I hope you can see my cursor, so this is an area that is in one of the developed classes, but it actually has quite a few trees in it, which doesn't, pop out, it's not as evident necessarily from looking at this map. And, I guess the other thing I would just point out is there's clearly a lot going on here. You can see a lot more in the very high resolution image than gets picked up in the classification, which is quite good. But, there's, the thing I want to emphasize is there's a lot of variation out here. on the landscape. And we can capture a lot of it, but not necessarily all of it, with these satellite-based land cover products. Just to point out a couple of other ways of looking at, looking at the data, we can look at continuous tree cover. So for example, we can see that we have some areas that are very dense forest, others where we have sparser cover of trees mixed in with roads and buildings and other sorts of things. And these are actually some of the base products that are used to make that classification that I saw you, that I showed you. But when we look at land cover through the lens of a satellite, these are the types of things that we're often looking at. And what I want to show you here, this is a flow chart that I put together for a recent paper that I wrote with some colleagues. And this was done specifically for malaria, but I think, and malaria transmitting mosquitoes. But I think it's generalizable to other mosquitoes and mosquito-borne diseases as well. Trying to understand, if you look at the top, these are some of the things that we can detect from satellites. So precipitation, land surface temperature, things like greenness indices, water bodies, soil moisture. Here we have land use and land cover maps, and I just showed you some examples of those. But sometimes we map things like population density, or we may try to map individual buildings or building density. And these, so these measurements can tell us about a variety of phenomena, like climate, for example. Land cover, land use maps, population, buildings, they tell us a lot about land use. So things like, and that's how, not necessarily the cover of the surface, but how are people using the land? Settlement patterns, types and qualities of housing, activities like agriculture, irrigation, we could put forestry in here too. And these affect mosquitoes and mosquito-borne diseases through a variety of pathways. One of the ones that we tend to think about a lot is mosquito habitat. We think particularly about the breeding habitat for mosquitoes. So temporary water bodies, potentially also the vegetation that serves as a resting habitat for some mosquitoes. So we can have these habitat effects. But land cover and land use, humans respond to that as well. So it tells us something about human living conditions and activities. Are people exposed to mosquitoes through, through outdoor activities? Is there seasonal migration for work in other areas? Do people have to travel to maybe malarious locations to work in agriculture? How close are people to healthcare? What is the potential for public health interventions? And so the point that I want to make here is that we've got we have these things we can measure at the top, and we have multiple and potentially rather complicated pathways through which again, what we see through the lens of the satellite tells us about things like mosquito habitats and land use and what humans are doing and what the climate is like. And that, those things all influence mosquito populations and the potential to transmit disease in a variety of ways. And then one last thing that I just want to share here. So when we think specifically about the mosquito habitats, these are often some of the specific things we're interested in. Depending on the context, it might be abandoned swimming pools, tires, and other refuse. It might be, borrow pits used in construction, or irrigated agriculture, either irrigation channels, rice paddies. And these things aren't always necessarily evident in the satellite-based land cover data that we work with. So, again, making the challenge, going from satellites to specific local features to mosquitoes and mosquito-borne disease risk. That's part of the research challenge. So let me kind of get right into it here. So I actually lived and worked in South Dakota for a long time. South Dakota is one of the hotspots actually for mosquito-borne disease transmission in the US, particularly West Nile virus. So that's something that we're very interested in. There are two very important mosquitoes and very common mosquitoes in South Dakota. One is Aedes vexans. It's a floodwater mosquito. Like other mosquitoes in the genus Aedes, they tend to lay their eggs in humid places, and you'll get big simultaneous hatches when these areas flood. And it is not an important disease vector. It's a nuisance mosquito, but it's a very common mosquito. Culex tarsalis, the Western encephalitis mosquito, breeds in standing water. So they actually lay their eggs in water, and they breed in a variety of natural and anthropogenic habitats, although usually not floodwaters that have high organic content. And that's the important mosquito from the standpoint of public health and transmission of West Nile virus. So these are some data from a publication that my postdoc Ting-Woo Chong wrote a number of years back, I guess about 10 years ago now. And so these are mosquito traps in mosquito surveillance data in, there we go. These are mosquito data that are being collected over 4 years. And here they're just summarized in terms of the mean count of mosquitoes per trap night. So we can see, bigger circles are where there are more mosquitoes. Smaller circles are where there are fewer mosquitoes. We have Aedes vexans on the left and Culex tarsalis on the right. And one of the first things you see when you look at the data is it's kind of noisy. We sort of, it looks maybe like with Aedes vexans, we have some bigger circles in, closer into Sioux Falls, which is the red area there, the city. With tarsalis, it sort of looks like we might see some bigger, larger circles outside the city, but it's difficult to tell. So we need to get in here and crunch some numbers to look at these relationships. And I'm going to skip over the details, but these are kind of the key results. If we can derive some standardized numbers that tell us how strongly each one of these land cover types is associated with each species, it turns out that the associations are really, really different. Aedes vexans, we tend to find in wetlands and not so much in grasslands. So the wetlands in this area, those are going to be lower lying, very flood prone areas, which again makes sense for a floodwater mosquito. Culex tarsalis, we tend to find them outside of urban areas and particularly in grasslands, not wetlands, and not in croplands. So, grasslands are, open. They don't have a crop canopy over top of them. They're open. Sunlight comes in, which is important for the breeding of Culex tarsalis. And we tend to get smaller, more persistent water bodies in ditches and other areas that are strongly associated with Culex tarsalis. And it turns out that those relationships with between land cover and Culex tarsalis also hold when we look at human West Nile virus cases. So we tend to find higher West Nile virus risk in grasslands at lower elevations and on poorly drained soils, lower in forests, higher elevations, and in cities. So we can see, it really pops out in the city of Sioux Falls here. We've got relatively low risk of West Nile virus relative to the number of people inside the city. higher in the surrounding landscapes, particularly in the western portion of that landscape. And, we tend to see blue within the black lines, the city of Aberdeen too, as well as Rapid City. And then in Rapid City, if you look to the west, we have the Black Hills. So we're going up into higher elevations. We have a lot of tree cover. We have there's much lower risk of West Nile virus transmission there than in the Great Plains. And so, and that was another study that Ting Wu, my former postdoc, did. One of my grad students, Andrea Hess, recently kind of built on Ting Wu's work to build a statewide West Nile virus risk map based on a combination of different things. Elevation, humidity, land cover comes out in here again. We talked about grassland associations before, and if you look here, we actually find strong associations with areas of pasture land. Also, some climate and soil variables are important. And just to point out, this wetness index is kind of a year-to-year variation. What it's really doing is measuring year-to-year variation in surface water and surface moisture. So it's telling us something about where the most flood-prone areas in the state are. And again, I'm not going to go into the details, but Andrea was able to put all of these pieces together and come up with a very nice statewide risk map for West Nile virus. And I would just point out that these three rectangles, Aberdeen up here in the north, Sioux Falls in the east, Rapid City in the west, those are the same kind of micro landscapes that I showed you before, but you can see some of the same patterns. We can see the city of Aberdeen here, coming out, and this is one of this northeastern part of the state. From a regional standpoint, is one of the biggest hotspots in South Dakota. But we see that the city of Aberdeen actually, like we saw before, has a lower risk of West Nile virus than the surrounding rural areas. And again, that's linked to the distribution of the main vector mosquito, which is more associated with rural habitats than urban habitats. All right, so let me move on now to the second vignette, and let's talk a little bit about wetlands and malaria in the Amhara region of Ethiopia. So this is an aerial photograph of the Amhara region, and it kind of gives you, I think, a lot of the main features of this region. It is mountainous, very rugged. It has an extremely dense, primarily rural population. Pretty much every scrap of arable land is being farmed. So very heavy human footprint, extensive agriculture, densely populated. And so what we were interested in here is understanding how the distribution of land cover and potentially some of the wetlands in this region. So anytime when you have, I guess the other thing to point out about this part of Ethiopia is it has huge amounts of rainfall. It's a big chunk of the Blue Nile Basin, what's called the water tower of East Africa. So you have a lot of water coming in, you have mountains, and then you have a lot of flat areas as well. And so that tends to create wetlands. We have a number of places where we tend to get, long-term seasonal inundation that creates mostly herbaceous wetlands. They tend to be too wet to farm, so they're often used as seasonal pasture lands for cattle. So we did a study where we collected, and this was my PhD student, Alamayu Mideksa. He collected a lot of training data at some of the sites that are highlighted in the map on the right. And he went through kind of a standard land cover classification process where he collected his, let me me see, collected his training data. Then he brought together both Landsat, so spectral data from Landsat, in addition to some topographic indices from a digital elevation model, put them into a machine learning algorithm, random forest, generated a classification, and then we validated it on some independent data. So a pretty standard approach. The things that I want to point out here are we got pretty high accuracies for the most part, for 80 to 90% accuracy for our different land cover classes. Just one point, if you look at these bars, when we used a combination of satellite data and topographic variables, we actually tended to get the highest accuracies, particularly for wetlands. So you see these dark green bars are the highest. And again, that makes sense because that topographic data gives us a little bit more information about where wetlands are likely to occur. So, if you see something in the satellite spectral data and it looks like a wetland, it's really likely to be a wetland if it's on flat terrain. If it's on a steep slope, it's probably not a wetland. So having that additional information helps us. And this is the regional map that we got on the left. And you can see that the wetlands, we have big regional concentrations of them. There are some alluvial plains here around Lake Tana. We have a lot of wetlands here. And then this is a big flat area, a flatter area on the slopes of the Choka Mountains. And then we have an escarpment where it drains down into the Blue Nile River, which runs along the southwestern border here. But here we tend to have some flatter areas where water draining from the Choka Mountains collects and creates some of these big wetland complexes. Just here's a picture of what some of these look like. So just to give us, so the yellow in this case is cropland. And, going back, like I pointed out before, most of, historically, a lot of this would have been forested, kind of temperate subtropical Afro-montane forest. But, historically, a lot of that's been cleared for cropland. So we tend to see a lot of these cropland wetland mosaics. And then this is some high resolution imagery if you kind of zoom in to the red box here. So here we can see some wetlands around a water body. Here you can see them fairly well. So these are, you can see the croplands, the fields, which are the yellow. And then these are some big wetlands. The local term for these, they're called musks. And again, they're often used as, they may have like knee to waist deep water in a lot of them during the rainy season, but then during the dry season, they may still have a fair amount of water in them, but they're used quite a bit for, as pasture for grazing cattle. And again, this gives you an idea. This is just a kind of a 3D rendering of the land cover map to give you a feel for the terrain. So again, you tend to see, and in this case, I apologize, this is a different color scheme, which is a little confusing. But here, the light green are the wetlands and the brown are croplands. And then you can see the dark green are trees. Those are often planted eucalyptus or kind of shrubs on these cliffs. So here you see the strong relationship between terrain and land cover. We tend to see a lot of wetlands down here in this flat plain of Lake Tana. If you look up that mountain, you can see that there's kind of a shelf up here where again, water can back up and you can have more wetlands up here. We could also zoom into one of these areas. So this is what it looks when you're in one of these. You can see some cattle in the background. This is actually right at the beginning of the rainy season. So it's kind of, it's kind of ankle deep water we're squishing around in here. But you see, because they're grazed, you can see that they're peppered with these hoof prints that hold water. And you can find a lot of Anopheles arabiensis, which is regionally the most important malaria vector in those hoof prints. So here's the situation, going back to some of the things we were talking about before, so you're not going to see hoof prints from space. But if you can see, the wetlands, the seasonal, herbaceous wetlands, seasonal pastures from space, you're able to identify the places that are likely to have a lot of hoof prints in them and potentially provide a lot of mosquito breeding habitat. And then we actually analyzed this with, we had some limited data, historical data on malaria cases. And you can see, so this is broken down by what are called waridas or districts. And if you look at the upper left, subfigure A, so the bluer areas have more wetlands. And then the other maps are dry season malaria, wet season malaria, and then the fall season right after the wet season. That's the traditional big malaria epidemic period after the rains stop. And there's a lot of water out on the landscape, but it's warming up and conditions are improving for mosquitoes and for disease transmission. And you have to kind of squint your eyes a little bit, particularly because we don't have data for every, or we didn't have data for every warida. But what we found was that districts with higher coverage of wetlands indeed had higher malaria prevalence. And when we compared it to some other variables, typically if you're trying to study, if you're looking at the geography of diseases over large areas, your go-to variables are things like temperature and precipitation and elevation. So here in the mountains, there's typically like an elevation cutoff, above which it's too cool for the mosquitoes and for malaria transmission. Here, though, interestingly, we actually found out that these wetlands, this land cover variable, was a better predictor of malaria distribution than temperature, precip, or elevation. So that was an interesting finding. And it was good to know that we could get some worthwhile new information about malaria risk out of this land cover data set. And let me finish, I've got one more example here. And this is actually a recent study that I did looking at some US cities, or looking specifically at one US city to assess suitability for the transmission of dengue fever. So, this is something that many of you are probably familiar with. This is a viral disease, has very severe symptoms. You can get, particularly if you're reinfected with it, multiple strains, you can get dengue, hemorrhagic fever, which is life-threatening. But in the US, we have two potential dengue vectors. There's Aedes, the big one is Aedes aegypti, the yellow fever mosquito. It's a very efficient vector, feeds mostly on humans, strongly associated with human habitats. Fortunately, we don't have a lot of Aedes aegypti. It tends to be found in high abundances only in Florida and other places that are pretty close to the southern border of the US. Another potential vector is the Asian tiger mosquito, Aedes albopictus. So it's less efficient, but it's driven some major outbreaks, particularly in southern Europe. And it's different from aegypti in that it feeds on humans and a variety of other animal species. And it's actually found in a much more diverse array of urban and rural habitats. And it's much, much more abundant and widespread throughout the US than Aedes aegypti. So it's something of a concern. Now, what we did here, and I'm not going to have time to get into the details, but there's been a lot of work and a lot of interest on climate and mosquito-borne diseases. And one thing that people have done is they've done a lot of laboratory work and compiled a lot of data from experiments to really understand how temperature as a driving variable influences the life cycle and disease transmission by various mosquitoes. So here, if we look specifically at Aedes albopictus, these curves tell us something about, okay, Aedes albopictus tends to bite, is most likely to bite, most active at about 30 degrees Celsius. On the other hand, it doesn't have a very long lifespan at 30 degrees Celsius. It tends to live the longest at slightly cooler temperatures. Temperature also affects things like the infection and transmission probabilities, and then what's called the extrinsic incubation period, essentially how long it takes an infected mosquito to be able to transmit the virus, to be infectious. And without getting into the details, so there's a lot of data on this, and people have figured out ways to put these into mathematical equations and come up with calculations of things like vectorial capacity. So if we crunch through all the numbers, and we know what the temperature is at a particular location, we can figure out from all of this the vectorial capacity. Okay, so what is the total number of infectious bites that would eventually arise from all the mosquitoes biting a single infected human in a day? So, and that's, if you don't quite understand all the nuances of that, it's fine. But it's essentially a number that tells us how rapidly are we going to be able to transmit that virus. If vectorial capacity is high, we have the potential for an outbreak. If it's really low, even if someone shows up with dengue and is bitten by a mosquito, it's unlikely that it's going to be transmitted rapidly enough to actually spread. So that's pretty cool. We've got temperature. We can put it through these equations and estimate vectorial capacity. And people have done that. People have done some really cool stuff with this. These are, so this is the potential for dengue transmission in the Western Hemisphere. Left, this is by, I believe this is by Aedes albopictus. And I've got to go back. Actually, this might be dengue and Zika. I apologize. I forgot to label it, and I honestly can't remember which one this is now. But the idea is that you can take these gridded climate data sets, and run these temperature values through these models and predict, based on this real kind of bottom-up mechanistic modeling approach, where is there a risk of disease transmission? Now, one of the things that's been pointed out is that, a lot of these data we tend to get from weather stations or interpolated weather data. But in general, most of the mosquitoes that transmit diseases are not going to be hanging out at weather stations. They're going to be, maybe in someone's backyard, which is, be close to trees and buildings, which is where we explicitly do not put weather stations. So we were interested in the question, so if we look in a city and we look at variation in the microclimates of that city, how important is that? Does it make a difference? And so what we did, and this is published in this PLOS Neglected Tropical Diseases paper, is that we had data on mosquito abundance. We have kind of coarse scale gridded data, climate data from GridMet. We have microclimate data. And this was all, and I have to say, this was data that was collected by my colleague, Courtney Murdoch, who was at the University of Georgia for a while. She's at Cornell now. But she and her lab did a lot of the data collection. So she collected mosquito data, microclimate data, and I did some land cover mapping with some sentinel imagery and very high spatial resolution imagery. So what we did is we put together this coarse scale gridded data with the microclimate data and our land cover data to essentially downscale the climate data to take into account the land cover effects on microclimate and come up with a 30-meter microclimate data set. And we combined this with and we use this to help us estimate fine scale mosquito abundance. put it together with some of those trait relationships into a vectorial capacity model. And I'm not going to show you this part, but then we also did some vulnerability assessments as well. So let me, again, as with a lot of things, it makes more sense if you look at the pictures. So this is the study area. So this is Athens, Clarke County, Georgia. So we've got population on the lower right. This is a Sentinel false color composite. And then the important things here are tree cover and impervious surface. So these were the main land cover data sources that we worked with in to downscale the microclimate data. Here's some of that meteorological data. So this is data from the mosquito season in 2018. So this dashed black line are data from the UGA weather station. So kind of right in the middle, essentially a rooftop in the middle of, right in the middle of Athens, close to downtown. And then if you look at the red and the orange, that is, oops, I'm getting ahead of myself here. So that's the range of microclimate temperatures that we sampled at these 52 locations. And so the point here is that the microclimate temperatures are much more variable, and in some cases, for minimum temperature, tend to generally be cooler than what we get at that one weather station. So, and I'm going to skip over the details here, but we use some fairly simple linear approaches to basically predict at a pixel level minimum and maximum temperatures as a function of GridMet data, tree cover, and impervious surface cover. And this is what that looks like in pictures. So the GridMet data is on the bottom. So that is daily meteorological data that we can get. And this is from, I forget the day, but this is like 1 day of data in July, I believe. And when we downscale it, you can see that we start to pick up some land cover related patterns that you can't really, that you just can't see in those coarser gridded data sets. If you look at the minimum temperature, that's essentially responding to an urban heat island effect. So, you know, that minimum nighttime temperature is going to be warmer where you have large concentrations of impervious surface. On the other hand, if we look at maximum temperatures, they are very sensitive to shading from the tree canopy. So, you know, again, if we're looking at it from the perspective of a mosquito, we've got, you tend to have higher temperatures in open areas and cooler temperatures where you have patches of trees. So, you know, so we're looking at, again, we're trying to model the potential for dengue transmission as a function of temperature, but to understand the temperatures that the mosquitoes are likely to actually experience in different areas, we need to take into account land cover and its influence on these microclimate patterns within the city. And let's see, oh, I'm running a little long, so I will just point out, so this is our relationship between, again, microclimate temperature and mosquitoes. So here we've got minimum temperature, maximum temperature. The size of the dots is proportional to the number of the mosquitoes. And we have it kind of a smooth response surface here. So the redder it is, the higher the mosquito abundance. So we tend to find these abundances within kind of a maximum temperature range of about 25 degrees Celsius up to maybe 33 degrees Celsius. So it's a unimodal relationship there. You can see that those dots get smaller as we start to get up to about 35 degrees Celsius. It actually gets too hot for the mosquitoes during the day. And there's a general increase, a unimodal increase in mosquito abundance with minimum temperature. So we can say, yeah, okay, there is, you know, we do find relationships between temperature and mosquito abundance at this microclimate level. Just quickly, this is looking at results for 4 different months of the season. And the story here is that if you look at the blue histograms, those are the histograms of vectorial capacity during each one of those months. And these are, we've relativized these VC estimates. So the numbers themselves aren't all that interpretable, but basically higher is a higher probability of disease transmission. And we compare those to the vertical lines, which are, those are the estimates at that UGA weather station. So the story here is that, that weather station is actually not all that representative of the actual microclimate conditions throughout the city. And if we were to depend just on the data from that weather station, we'd probably, actually be overestimating the risk of dengue transmission. Now, when we map it, this is what it looks like. So the red areas are essentially the hotspots, high vectorial capacity. And if you look at them, and well, you probably can't tell unless you know Athens really well, but you look and we kind of see these red, like the reddest areas are in these little clusters or islands, and they tend to be more into the center of the city, particularly in, from June through September. And so what we're seeing there is those are actually areas that are within city limits in that developed central core of Athens, but that have tree cover. So they're kind of green islands within the city. And just to sum it up, what we found was that if we kind of look at this stylized gradient from rural areas on the left and on the right to kind of the urban core in the center, that minimum temperature comes up at night because of the urban heat island effect. Where we have tree cover in the city, that brings our maximum temperature down. So we're actually compressing that diurnal range of temperature, but we're compressing it within the optimal range for dengue transmission in this particular context. So, in this particular context, it's not necessarily going to extrapolate everywhere, but that combination of a lot of impervious surface and having some patches of trees in there is actually creating the most favorable microclimates for dengue transmission in the city. Now, we don't, again, this is all potential because we don't have active dengue transmission in the US and certainly not in Athens. We used Athens because we had a really great data set that Courtney had collected. But anyway, so that's kind of the take-home message of that story, is that we tend to think about land cover as being important for mosquito habitats, particularly breeding habitats, but microclimate effects are potentially important as well. And I know I'm running a little bit long, but and hopefully people can, you guys can send this out to the attendees. These are, for people who are researching this, these are a couple of good sources of land cover for the US. This viewer for the National Land Cover Database, I use it with my classes all the time for their projects. I think it's the best way to grab land cover data, NLCD data for the US, or even if you just want to browse it and look at it. And I'll just mention LCMAP is a newer and somewhat different source of land cover data for the US that is, again, I don't have that much to say about it. It's new, it's different, it goes back further in time. It's an annual product. It's a little more focused on change detection. But if someone's working with land cover in the US, it's worth checking out. I apologize, I don't have international links here, in part because I kind of feel like there aren't that many, there isn't what I would consider like the canonical global land cover data set that I would recommend for everyone to use. So I apologize, but if, I could give some suggestions if people are interested in particular areas. And then, again, I don't want to spend a lot of time on this, but, just some lessons learned for anyone who's on the call who wants to work with looking at land cover mosquito relationships. I think one of the things that you have to deal with is the relationships are always going to be noisy no matter what. So, if you want to look over a really broad range of land cover characteristics. It's going to be hard to find a signal if you've got like 10 sites that are all downtown in your city. But you can often pick out, rural to urban gradients for different mosquito species. Often I find that working with the continuous variables like percent tree cover or impervious, they're a little more predictive sometimes and can be easier to work with in categories. This is something I always tell my students. Don't overlook, basic visualizations, simple statistics, correlations before you go stampeding to put everything into, a big machine learning algorithm and see what comes out the other end. Incorporate local observations of land cover and land use too. If you're out there collecting mosquitoes, you can see those details of land cover. You can see where there is, trash that might be create artificial breeding sites. You can see where in your neighborhood water tends to pond. So factor that in. Again, take species into account. And then the other thing, people often get disappointed if they don't get some, 99% predictive model. But, because land cover is one of many things that influences mosquitoes, don't necessarily expect to have incredibly high accuracy. And I would even say, if you do get some model that makes an almost perfect prediction, you're probably doing something wrong. So anyway, those are just, as with all free advice, take it for what it is, but that's all I have.